Hello, everyone. Lots of people are joining the line right now, so we're just gonna get started in 1 minute or so while people are still kind of popping on. But we've got a great call today, so we will get started. You don't want to waste too much time. I'll go ahead and open while we have more and more people joining the line. It does seem like we've got kind of critical mass, but just to say welcome. My name is Stacy Hanna. I'm the Director of Research Engagement at AVAC, and I'm really excited. This is our third and final HVAD webinar for the year. We've had a great series and kind of closing out the month of May, closing out the kind of the month of HVAD here with this sort of final call. This call is going to be it's going to be fun. Those of us on the line have already been having fun, and we're going to be talking about we're going to be kind of looking at the sort of the fourth P in the series, so prospects, and I think having some thoughtful and provocative discussion about the future of HIV vaccine research and development. Just as a reminder for those of you who did or who didn't tune into the first two in the series. The first call talked about the pipeline and platforms, so looking at mRNA specifically and sort of where we are with the current state of vaccine R&D. So a really nice overview there. The third P was processes. So we looked at kind of some new approaches in terms of clinical trials around experimental medicine, and also PrEP with this sort of combined PrEP and vaccine regimen. And again, this is where we're really going to kind of bring it all together. I think on our first two calls, we didn't have a ton of time for discussion, and we have had questions coming in both on the calls as well as in separate emails that we want to kind of get to here with our panelists. And so I'm going to pass to my co-moderator, who's going to introduce our panelists, but I'm joined by a very great colleague, Ethel Makila, who's the Director of Policy Advocacy and Communications based at IAVI Nairobi. And over to you, Ethel.
Thanks so much, Stacy. As you said, we have so much to talk about, so I'm not going to take too much time. I think I'll just rush over to introduce our very great panelists. I have the absolute pleasure to introduce our panelists for today. Dr. William Kilembe, he's the project director for the Center for Family Health Research in Zambia. He's also a principal investigator on several IAVI studies. There's also Galit Alta, principal investigator at Ragon Institute of Massachusetts General Hospital, MIT, and Harvard. We also have Dale Hu. He was previously the Vaccine Clinical Research Branch Chief with the Division of AIDS at NIAID. He now serves as a consultant to the Vaccine Research Program at DAIDS. And last but not least, we have Bill Snow, who is a co-founder and a senior advisor to AVAC. So I'm going to hand it over to Stacy to get us started. Stacy, back to you. Okay,
thanks a lot, Ethel. So we did say kind of no presentations here for this call, but we did want to just start out with just a couple very brief, very kind of high level slides to just sort of set the scene in terms of where we are in the HIV vaccine kind of R&D landscape, and then we'll quickly pass it over to our panelists, who will really kind of give their perspectives on where we stand. And I think so. I'll just kind of jump into this next, to sort of the first slide. I think that these are details that everyone on this line are probably quite familiar with, but again, just to really kind of set the stage, we wanted to sort of look at what's happened in the past 2 years. I think everyone knows the past 2 years have been really kind of a wild ride in research across the board. Not going to get into everything on this slide, but just wanted to sort of highlight what we see here, specifically around HIV vaccines and BNABs and the efficacy trials that we've seen read out over the past couple of years. Again, everyone is probably quite familiar with this, but just to review, in January 2020, we saw the early stoppage of the Uhambo trial. The Uhambo trial was testing the candidate that was based on the RV144 vaccine, sort of adapted and tested in men and women in South Africa. The trial did end early because unfortunately it just showed no sign of efficacy at all there. And then a year later, we saw the readout from the AMP trials. And again, everybody knows these were sort of two large efficacy trials conducted globally and in various populations. And AMP overall, and it was testing VRC01, the BNAb VRC01, as a passive infusion to see if it would prevent infection. Overall, as we know, the trial did show no efficacy across the board, but it did show about a 75% efficacy amongst people who were infected with HIV strains that were kind of highly sensitive to the VRC01 BNAb. And so people really see this as kind of a key step in terms of sort of the BNAb effect, passive BNAbs, or even eliciting BNAbs in a vaccine. Going into AMP, I think everyone knew that we would need a combination of BNAbs as a passive product, but I think what AMP showed us was that potentially the bar for that combination, what we would need was actually probably higher than we had originally kind of hoped or thought, but even with kind of VRC01. And obviously lots of work continuing on around BNAb development from AMP, but really sort of a key development for the field. And then finally in September, I think it was around September of 2021, we again saw the early stoppage of the Invacoto trial. This trial showed, again, kind of the point estimate was around 25%, but that confidence interval did cross 0. And so really kind of, again, showed overall no efficacy, but a lot of thought into potentially what this sort of trend towards efficacy was, especially in certain populations in that trial. And that trial was conducted in sub-Saharan Africa in women. And I think we'll talk a little bit about that as well, or maybe more about that in our discussion. And then just as this slide shows, I mean, all of this is against the backdrop, both of really this incredible landscape of COVID vaccine development over the past few years, as well as movement in ARV-based prevention, notably with the dapivirine vaginal ring, as well as CAB-LA, or CAB for PrEP, or injectable CAB, as it's now being called. So yeah, just reviewing kind of the landscape. And then these next two slides are just, again, a recap of, I think, some sort of, what we maybe see now as prospects in terms of vaccine research and development. This is a very detailed snapshot, and this all comes from a fact sheet, and I think someone can kind of pop the link to this fact sheet in the chat, because obviously I'm not going to go through all these details, but what it does show is that we've got kind of 3 ongoing trials of mRNA-based vaccines right now, clinical trials. So that's really exciting. A lot of focus on mRNA as as having lots of potential for HIV vaccine development. But I think a recognition that we do need to kind of temper expectations and really kind of understand that mRNA is just a platform, and we're really kind of trying to figure out what antigens we need to develop an effective vaccine. This table is also a little bit of a mashup because it also shows trials testing this approach called germline targeting that is actually, again, in very, very early stages of research, but it is showing some promise. And so I think something to really be watching, and we'll be watching these trials in the coming in the coming months, and really seeing sort of what promise there is for HIV vaccines, both in terms of mRNA as well as germline targeting. And then finally, a lot of focus around experimental medicine, this idea of smaller, faster, more iterative first-in-human trials that are not developing candidates to be necessarily to go through the whole kind of research and development or product development process for licensure, but instead are testing scientific hypotheses so we can actually kind of get to this, get to the bottom of sort of the scientific challenge of developing an effective HIV vaccine candidate. And so that is the end of the little little overview, but just again to kind of set the stage and to relieve our panelists from having to having to go through that on their own. And I think that I will just go ahead and kick off our panel discussion. We've asked each of our 4 panelists to kind of come on and to start off to give their perspectives on the current state of HIV vaccine research and development. And I'm just going to go sort of in order of my screen. And so, William, I'll start with you.
Hi, can you hear me?
Yes, we can, very well.
Thank you to AVAC for inviting me to this panel discussion at an important time in HIV vaccine research, especially after a time when we've received results that we didn't want to receive at this time, but in any case, we've received these results. But I think I'll begin by saying that the search for an HIV vaccine is challenged by many aspects, as we all know, and this is really something that we're aware of. One is that there's a lack of a model for natural immunity for HIV. It's a highly diverse virus, which has been necessitated and complicated by the rapid mutation of the virus. And also there's an inability for human to self-cure. So unlike other viral infections, HIV is a bit different in this way. And vaccine research and design will remain a process by which we have to learn what vaccine is going to work, which candidate is going to induce the immune responses that are desirable. And hopefully we can induce in the near future or in the distant future, we can induce immune responses that are better than human immune responses to the natural infection. There certainly is a lot of advance in the science, even with the results that have been received in the past 5 years. But what we need to realize is that this research really needs to go on. We need to advocate for this research to go on. There has to be quite a lot of political will and substantial funding to support the endeavor for HIV vaccine development. And also from the clinical trials that we've seen, we really see that an HIV vaccine is really important, because even in the clinical trials that have been conducted, when you look at the HIV incidences in those clinical trials, it still remains very high in those key populations, which actually explains the need and necessitates the need for us to continue searching for a vaccine. And so there are discussions in the field. I think there are newer ways of doing clinical trials that are being discussed. We've heard from the previous 2 sessions of models that can be used, including a cleverer way of doing clinical trials to try and reach answers quickly in using experimental medicine vaccine trials, and also trying to be smarter at how we induce broadly neutralizing antibodies through germline targeting. And I will not belabor that, but I think some of the panelists on this team that have better expertise will probably be able to elucidate some more information on that. So I'll end there for now.
Excellent. Thank you so much, William. Galit, next.
Thanks, Stacey, and thanks again for the invitation. It's wonderful to be on this panel this morning. So I was so excited when I saw the invitation to be here for one reason and one reason only, is that, we're in a moment in the history of vaccine development that is unprecedented, right? We have just seen more platforms, more compounds, more adjuvants tested than we've could have ever imagined, right, as vaccinologists that have been growing up in the HIV field over the last 20, 30 years. And so I think this moment really gives us a new rush and exhilaration and opportunity to begin to explore new grounds, really, go beyond the frontiers that we thought were ahead of us in the HIV vaccine space, and to really take a page from this, COVID experience to try to amplify and accelerate our ability to succeed and make a big difference in the HIV vaccine space. We discussed a lot of, Stacey, you raised a lot of the issues with the failures in the HIV vaccine spaces, but I would like to call those actual learning lessons rather than failures, and really, put the question in front of the audience and in front of the panel today to say, have we learned all we can learn from those experiences? And where can we move from those learning lessons? Because there were glimmers of hope, as you mentioned, right? There was that glimmer of hope in AMP that showed that there was efficacy against matched strains. There was a glimmer of hope in Invacoto, where we saw that there was protection, particularly in an age-dependent way, where there may have been a lower risk in particular populations. So to really ask the question, how can we maximize on those learnings while simultaneously, using this COVID success to really move us as fast as possible into this productive new direction? But I want to just raise one little question, just, from my perspective, and, and really try to ask, why was COVID so successful? Whereas HIV vaccines, we've struggled for so many years, right, to see these like glimmers of hope. And I think in my mind, there are really three reasons, if I can just kind of put those out there as kind of a controversial way to spark some discussion. The first reason, I think, of course, these are different viruses. And we know that HIV infects and establishes lifelong infection. It is incredibly evasive. It is highly mutable, as we just heard from William. But beyond that, it is a virus that has learned to subvert and eliminate our master orchestrator of all immunological function, the CD4 T cell. And so for that reason, we're dealing with a, really difficult target, a potentially, and I hope not controversial, more difficult target than SARS-CoV-2. And so we're going to have to break some dogma, and we're going to have to really challenge the notions of how we think vaccine development has to proceed in order to really counteract and come up with creative solutions for this particular pathogen. The second thing I just want to mention is there were more trials for COVID-19 in like 6 months than there have been in 40 years since the development, since like the discovery of HIV. I mean, that to me is just absolutely unacceptable, right? I'm saying it with a big smile on my face, but I should have, a big frown. But the fact that we were able to test so many hypotheses so quickly and learn so much about the immunological response at light speed in this COVID-19 pandemic, I think is a lesson that we have to bring to the HIV field. And like you said, Stacey, experimental medicine, vaccine trials, where we can really begin to learn how we can, tune the immune system through these different platforms. That I think is one of the biggest, differences between the two groups. And the last thing I want to mention, I think I mentioned this before, but the last difference I just want to highlight is the fact that, in the COVID-19 pandemic, we went in relatively undogmatic, right? We did not presuppose that we knew exactly how the immune response fought this pathogen, but we tested all these concepts, exploring different platforms, different compounds through different kinds of vaccine strategies with that express goal of essentially allowing the immune system to teach us how to make the best intervention for that particular pathogen. And so I think that it's time to drop the dogma, right? And there has been a lot of dogma that has, really obscured a lot of the vaccine development efforts in the HIV vaccine space that I think that we need to begin to kind of shed as we move into this new era where we have the capability to really use these new technologies to move the needle in a completely new way and to end the issues that this virus brings on. So I'm going to stop there.
Incredible, Galit. Thank you so much. I just wanted to say, I don't think I said the word failure. I don't never say failure. Okay. It's my failure.
It's my personal, yeah.
Success.
Okay, yeah. Not a failure.
But thank you for that, and lots of great stuff that we'll keep talking about. So next we'll go over to Dale.
Thank you, Stacey, and thank you, Bethel. Thank you, Mitchell, as well, for inviting me this morning. I actually the order that you gave is really nice, because I do want to build on what William and Galit said. I think in a lot of ways with COVID, we were incredibly lucky that we had a virus that was amenable to rapid vaccine development. And as Galit said, HIV is really kind of an amazingly challenging pathogen. The other point that I think is really, I want to build on what Galit said, is I think that there are a lot of learning lessons. And one thing that was very valuable from COVID, again, was the huge amount of safety data from mRNA. And that will really give us a lot of advantages in moving forward. mRNA obviously has a lot of advantages over protein. You can iterate faster. I think the time for revamping different proteins is much more expensive and time intensive. I did want to also bring up kind of a perspective that that I've brought up before, but now that I'm not a government employee, I feel like I have a little more freedom to actually advocate for the government. I mean, I think the federal government really had a huge role in the COVID response. And I just want to jot down a couple things that I think put things into perspective. J&J, Moderna, Sanofi, and AstraZeneca are estimated to have received more than 2.7 billion from the federal government to cover expenses related to clinical trials. In comparison, on May 10th, the Biden administration announced 40 billion aid to Ukraine, in addition to the 2 billion since the beginning of the Biden administration. Not to diminish what's going on in Ukraine, but just to compare that here we're talking about a worldwide pandemic affecting basically almost all countries. Now, from the DAIDS and NIAID perspective, we're looking at a much, much smaller amount of funding. And here I kind of feel like no longer as a federal employee, I feel kind of more free to advocate for increased funding for research. So an example is DAIDS funding for the HVTN for 4 years between 2014 and 2017 was about $250 million. Direct funding to the CHAVI for both CHAVIs actually was approximately around the same amount, 250 million for 8 years between 2005 and 2012. That amount of funding is equivalent to 1 Patriot missile battery. So when you compare that to DOD, again, DOD is very important, but just to show the difference in magnitude that we fund HIV research. The direct costs for RV-144 were $115 million from DAIDS. The direct costs for 702 were $131 million. This is about the same order of magnitude for those of us who are Washington Capitol fans. Alex Ovechkin's 13-year contract was $124 million. So, you know, if again, not to demean that, but when we put these in comparison, we really are under investing in HIV research. I want to add 2 more comparisons. So the direct cost for both AMP studies was $144 million. That was less than the cost of 1 F-22 Raptor fighter jet, which is $150 million. The 5-year funding for both Duke and Scripps CHAVI IDs was $190 million, which was less than the $200 million to produce the film blockbuster, The Titanic. I just want to end there by saying that we are severely underfunding research for HIV, whether that be basic research, translational research, clinical trials. And I'll just stop there.
Wow, excellent. I think we'll have to incorporate some of those comparisons into our resource tracking report. So that definitely puts it in perspective. So yeah, thank you. So last but not least, we will hear from Bill.
Thank you, everyone who came before me. Dale, you were unleashed, unbound. It's amazing. It's interesting to see what happens when someone gets released from government service. But all of you have been great, and you covered most of the points I was going to make. So I'm going to get personal for a minute. I've been engaged with HIV vaccines as my main advocacy cause for more than 30 years now. And on the first of these calls, I said we needed to triangulate between what we were going to hear on these three calls. So now is my opportunity to triangulate. I've been trying to write what I know and what I understand has happened in HIV vaccines. And it's so technical and so broad and so complex and so completely unresolved, as Galit has mentioned. We're in a world where we finally have a licensed malaria vaccine after 35 years. Of course, we've talked about COVID a great deal. It taught us enormous lessons about the downstream effort of getting a vaccine out into the world. But there was a time when HIV was the zoonotic disease of choice, right? It was the horrible thing that came from animals that was killing humans in disproportionate numbers. There's going to be more. It's not a surprise that HIV has taken somewhat of a back seat, but it is still infecting more than 1 million people a year. So it's important for a new generation of people to be caring about HIV. And the last 2 years have been really challenging because we really don't understand what happened. And the reason we don't understand is because we thought we knew the answers. Galit said that very thing. And immunity is a system. It's not a single thing. It's developed by evolution over millions of years. It's a kludge. It's stuff getting added on to other stuff. And it's more complicated than people. I once said to Bonnie Matheson, well, gosh, cells are as complicated as people are. And she winked at me and she said, oh, more complicated than that. So we know that AMP has set a high bar. We know that it's not the whole picture. We know that there are other people doing interesting things. There's a group at NIH led by Paul Lusso that's making virus-like particles that are spitting out tier 2 antibodies and cross-clade and protecting primates. There's people like Bruce Walker who are reminding us that the immune system has at least 2 arms. There are people like Galit who are reminding us that the whole antibody function and innate function have dozens of mechanisms that could help. So yes, broadly neutralizing antibodies are important. They're the most direct, clearest way to get protection, but it's going to be hard. It's going to take combinations, and no one has ever done it before. So, so the real question is, how do we speed up getting an HIV vaccine? And I think we'll be talking about that for the rest of the hour. Thank you.
Wow, amazing insights from all of you. My mind is just buzzing. I have a new slogan, drop the dogma, from Galit. And somebody has actually asked a specific question to you on that, Galit. But before we go further, I just want to remind us that there's the Q&A section in the bottom of the screen. It's labeled Q&A, and you can post your questions there. You can post them anonymously, or you can include your name. And speaking of posting questions, there's one very thought-provoking question that came to us before we launched the webinar. And I'm going to give it over to Stacy to share the question. It came from one of our advocates, very thought-provoking. Thank you so much for having shared your question with us ahead of time.
Great. And actually, this question links to, it's very similar to a question that just came in for the host and panelists. And it really is around this idea. We would love to hear your perspectives on kind of projecting into the future, right? We know at this point, we know we're not going to have an effective HIV vaccine for a long time, and no one ever likes to predict the number of years, but let's say, at least 5 years, probably 10 years, potentially more. And we know that the prevention landscape is changing as we speak, right? And projecting out, that far into the future, it's going to look even different. And this advocate, wrote in this question, and I think it's something that's on all of our minds. And I think that, we all know that ultimately we do need a vaccine to end the epidemic. But this person, put it so well in saying, 10 years from now, looking at the landscape, will people care about a vaccine anymore? Especially if an HIV vaccine requires 4 to 6 shots or, kind of regular boosters as we're seeing in COVID. And again, we know COVID and HIV vaccines are entirely different. But again, just thinking about what is in kind of lay people's minds right now, especially if there are more and more kind of ARV-based options out there and longer lasting, or options that people are liking and using and are working for them. So yeah, we'd just love to hear in any order thoughts about how will vaccines kind of fit into that landscape. Should we be looking now at kind of changing that sort of target product profile ultimately for a vaccine, kind of based on what we're seeing in terms of prevention, even things like the kind of the level of efficacy that we're shooting for. So that's a lot all in one.
Can I go for Stacey? I'll start off the answer. And so when I hear that question, I think about the grim reality that we saw roll out as vaccines for COVID-19, became available to the globe, right? We all thought, I think as a community, that these vaccines were, really our savior, right? They were coming to basically tip the balance of immunity on our side so we can battle against this virus that was incredibly unpredictable, although clearly not as lethal as HIV is in the absence of antiretroviral drugs. But the reality that I saw was, there were significant segments of the population that resisted this intervention. And if that's the case with a vaccine that requires 2 shots, maybe a third, we know that with PrEP and with other types of antivirals, long lasting, there will be segments of the population that will, resist, coming and, moving forward to take on those types of interventions. And it will be, we'll be surprised again at, which segments of the population may be more resistant, those that are unable to come, and get their PrEP, or just really not able to deliver to every corner of the globe. And so coming up with a second strategy, a strategy that still requires less of an intervention than coming in every 6 months or once a year or so on and so forth. I think that is, the nugget of hope that we can offer to the globe to really put up some defenses against this pathogen that really has a 95% death rate, right, if untreated. The game is completely different. And so I think that we need these backup strategies, no matter how good we are with prevention, no matter how effective we are at getting, phenomenally long-lasting drugs out there. We need to have that backup mechanism in place. And as William said, we know that the immune system can do this. We know that the immune system can self-cure. We know that the immune system can resist in some situations. And so just learning that immunology so we can make these different flavors of vaccines, right? Not the traditional flavors that we've used in the past, but these new types of vaccines that are easy to deploy globally. That is the backup strategy while we have this phenomenal therapeutic Band-Aid, is what I would call it, till we can get to allowing our immune system to fight the infection.
Thanks. Other thoughts? Go ahead.
If no one else is adding, I did want to add to what Galit just said. First of all, I think if we look back at 702 and 705, I think the original hope was that RV144 was kind of a minimum amount that we could improve on. And by changing the immunogen, modifying the adjuvant, the hope was that we would be reaching a higher upper range. And I think the disappointment there is that rather than that RV144 being, say, a lower range, it may have been not that. It may have been actually a ceiling, as our director at DAIDS, Carl Diefenbach, had mentioned. As Khalid and I think William mentioned earlier, I think the AMP studies were encouraging when antibodies are well matched to the virus. I think the other prevention strategies that are coming along, I think are really excellent. And the experience with both COVID, with HIV, other pathogens in monoclonal antibodies is extremely encouraging, I think, in that way. It reinforces the need for combo therapy. I mean, that's true for any infectious pathogen, whether it be antivirals, monoclonal antibodies, or even antibiotics. And I think that moving forward, we do have to consider how we incorporate these other prevention interventions within vaccine development. I think that the age of looking at vaccines as a standalone intervention is going to be difficult for many reasons in terms of the logistics of large trials, et cetera. I do want to mention one other thing that I think the experience that one example that I've thought of in the past, and before I had worked at CDC in the Division of Viral Hepatitis, early on there was a lot of kind of enthusiasm or effort to do a vaccine against hepatitis C. And it was similar to HIV and having a large amount of genetic variation affecting a lot of populations at risk. But when effective antivirals were developed for HCV, it essentially really kind of cut the knees off HCV vaccine development. And that's something we don't want to see happen with these other prevention interventions, because vaccines have huge advantages in terms of cost, if it's the right vaccine. All those things I think are important. And then following up also with the COVID experience, even if we have a 90% or over 90% effective HIV vaccine, which I certainly hope we can see in our lifetimes, if the uptake is 50% or less, then what good is that? And that again points to the importance of community engagement. And I think that was a lesson that the COVID vaccine trials really learned and built upon HIV, which I would say is kind of the gold standard in community engagement. And that is something that I think is a good lesson in terms of lessons both from HIV to COVID and from COVID back to promoting HIV vaccine development.
Stacey, I'd like to jump in if I may. I personally think that if we can solve the HIV problem, which is basically a breadth problem, a diversity problem, we're on the road to a universal vaccine. And we could figure out how to make universal vaccines for other diseases too. And I would just like to propose that even if we had long-acting prevention that people could use as an implant when they need it, we still might want to have, and I think that we would want to have a childhood vaccine. We'd want to have something that we could use where we have the time to prime people in order to make them resistant to HIV when they get exposed to it. And I also think that this bleeds into the cure question, because if we have a vaccine that works for prevention, we might also have a vaccine that would help us with ATI, with treatment interruption. and that would help teach us how to work towards a cure. So it's all coming together, and we really can't give up on this. It's essential that we don't.
I think I would just like to add to what the panelists have ably put up, to just say that indeed we know the models of vaccines for other diseases that have worked very well in terms of prevention of those diseases and in fact controlling them. And there is a great historical perspective of these sorts of situations where we've actually eradicated disease. And I think it is important that even at this time, when there are development of new strategies of vaccine development that show great promise, targeting highly conserved parts of the proteome of HIV, that are really directing how broadly neutralizing antibodies are actually generated, that it is key that the vaccine agenda actually progresses. And then the other aspect I would also just like to make is that the prevention strategies that are in the toolkit at the moment are, in my view, not very sustainable. They do prevent, but they require that someone receives a repeat dose, for instance, for PrEP. If we found even a long-acting PrEP strategy, it's going to take many repeat doses. Adherence has been a problem, even to Depo-Provera, which is a contraception that prevents pregnancy. And there are models where we see these challenges. The other thing is that when we start looking at cost effectiveness, we should never forget that an HIV vaccine, even if it is given, we found an effective one that is given maybe every 3 to 5 years, better still given every 10 years, is cost effective than actually administering antiretroviral based prevention strategies. Thanks.
Wow, thank you so much for all your responses to that question. And what I'm hearing is that, in as much as we do have other prevention interventions and potentially long-acting interventions, we still should be looking for an HIV vaccine. And that really brings me to the next question, which has been raised more than once, and which Galit alluded to. So we spoke about the not so successful or the efficacy trials, the trials that brought us results that were not what we expected or what we wanted, frankly. And have we really found out or learned all that we can from the previous vaccine trials? So Galit, you referred to them as lessons learned, learning lessons, as opposed to failures, trial failures. So have we really learned all that we can learn about them? Have we learned, and are we sharing this learning collectively? Or are we still, how are we responding to these lessons that we've learned from the past trials?
So Ethel, if it's okay, maybe I can kick this one off, and I hope that my co-panelists will argue against me. But honestly, I think, there was an extraordinary collective unity that occurred after RV144, right? I think we all experienced it, where there was that signal, we all came together, we sat in, we thought through all the possible experimental assays we could run. We, came up with a collective strategy that would allow us to pick the best in class assays so we could define correlates of immunity as quickly as possible. And it was all because we didn't cross that 0 threshold, right? But 0 thresholds are so arbitrary. I have to say, statistics are changing and evolving, as does science. And there were these glimmers of hope in all these, additional trials that have occurred, including the AM trial, where it was in the sub-analysis of the more conserved strains or mass strains, where we saw that signal, right, of efficacy that is, so incredibly important, because it tells us that neutralizing antibodies can, put up that barrier even in the human population. So we have not gone back in and done that for 702, and we've not gone back and done that in 705. And I don't want to blame it on the COVID pandemic, because we should not be blaming everything on COVID, right? It is our responsibility as a community that are committed to ending this epidemic, that we sit down and we say that it is our collective responsibility to begin to do this. But my argument would be is that there was funding fatigue, right? Dale showed it to us. He gave us all the examples of where that funding fatigue has played in and how absurd it is that we as a community have not felt that it was important enough to make that investment in digging in to those potential signals that are hidden within these trials. And it's that funding fatigue that has, not accelerated our ability to learn as quickly as possible from those trials so we could put more concepts into the pipeline, right? It is depressing, Stacy, that we have 3 concepts moving forward. That is, really, I think, unacceptable. I would say it's almost like horrific to think about the number of people that are, living with HIV in the world and people who are potentially, will become infected based on our projections. And we only have 3 concepts in play and they all test the same hypothesis. That is complete nonsense. And so it is our responsibility, maybe as a community, to really make that, very clear to the world that it is a small investment that we have to make that can, capitalize on all the incredible scientific accelerated, new technologies that we've developed over this last pandemic to really break that dogma and begin to create these new concepts that go into the pipeline so we can end this pandemic.
Stacey, I want to play advocate seriously now. Thousands of people volunteered for these trials. They gave their time, they gave their blood, they gave their hope into these trials, and we have not done enough to analyze what came out of that effort. The field as a whole is not looking to see what mistakes we made, what we might have missed, and they're just doing the same thing they've been doing for 30 years, jumping to the other side of the boat. Everybody's leaning in the other direction now because they're not happy with the result. And there have to be, like Khalid says, glimmers, or at the very least, we can put certain ideas to rest if we do this thoroughly.
Great. Do other panelists want to jump in on that? Khalid, and I wonder because I think that this really is such a critical issue. And actually, I think you're laying out really kind of an advocacy call to action, both of you. And so just wondering, are there specific things that can happen to make sure that this, data from these huge trials doesn't go to waste? How do we, are there kind of ways that advocates can support that? And also, can you kind of speak to any of the sub-analyses that might be coming out and how we can kind of be watching and advocating around those?
NIH should put out a call, or CAVD should put out a call to ask for ideas from researchers about what to do with these samples and whether or not we can figure out how to explain what went wrong with our hypotheses. There are so many differences between 702 and RV144 that we don't have a clue what dimension we erred in. And that's hugely important.
But let's also add in, Bill, right? We did not test the same hypothesis.
No, we
right?
didn't.
We tested a completely different hypothesis. We took, a trial that, showed us some level of efficacy in a low risk population with totally different inserts and totally different adjuvanting strategy. And we moved it to South Africa in the higher risk population, of course, where a vaccine, is absolutely needed, but we were testing a completely different question. Dale went through this also, right? And so, at least in the scientific realm, I would say that was a failed experimental design. But that doesn't mean we can't learn from that because, clearly there was still, some evidence that there was a V2 signal there, but have we dug in enough to understand what that V2 signal was? We're now seeing that there might be, a V2 signal also now with, Invacoto with the 705 trial. And so have we dug in enough and have we done the cross comparisons to really understand what is going on there in high risk, low risk populations, understanding across the age spectrum, really understanding, how these products, as, Bill was mentioning, are tuning the immune system in different ways and really comparing back to that original trial where we did see that glimmer of hope. And so I think that, we are rigorous scientists. We know how to do this work, but we have not been empowered to do this in a way that can hopefully create that, data that allows us to move forward. And how we do it, Stacey, I think there's a billion different ways, right? We have learned over this last pandemic, right, that people are very creative and lucrative in how they can, motivate, getting the funding that they need to do this work. But it does take a collective action, I think, from, everyone in the HIV vaccine development space to really say that this is a priority. And if we say that this is a priority and we agree that understanding this, so we can develop new strategies and really start thinking about, moving forward as opposed to standing in one place and wondering when is the vaccine coming. I think that really is something that we have to agree upon, and then find all the different, crowdsourcing, NIH, Gates Foundation, and other, organizations that are really passionate about this problem and feel that this is a problem that we can solve and must solve as a community.
So it's evident that we haven't done enough to learn from, or to take the learnings from what we have already experienced in as far as vaccine design and the trials are concerned. So what can be done to accelerate this process? How can we do this as rapidly as possible for vaccine development?
And maybe, Galit, you mentioned in your,
that's
to
to all of us. It's to everyone.
And I did want to put out this issue, Galit, because it prompted some questions in the chat. I'm trying to, there's actually a lot of chat, trying to keep track of it. But this issue around dogmas, and someone asked if you could kind of explain a little bit more. I think this is kind of what you're getting at, but would love, if you have any more sort of detail around what you mean by kind of dogmas, and would love others' thoughts on that as well. Like, how do we change the paradigm? We've also talked about, if people, because I know we're coming to the top of the hour, we've talked about this issue of kind of experimental medicine trials, and would love to hear thoughts from people about how we ensure that these trials are actually accelerating the overall process of getting to a vaccine.
Okay, let me do the dogma really quickly because I want to make sure that we can get into the trial piece of it. So I love what Roger put into the chat, because the field is like a butterfly, and it's absolutely true. I always think of it as a pendulum, but a butterfly is much better because it kind of jumps back and forth to wherever it sees like a pretty flower. And the pretty flower is, first, it was antibodies. Everyone was focused on antibodies, right? We came up with VAX-03 and VAX-04, drive, induce the highest levels of antibodies, and then we're going to get protection. And of course, we didn't see any level of efficacy because, just having more is not necessarily better. And that was a big lesson we learned that really forced us to start thinking qualitatively as opposed to quantitatively about the immune system, right? That swung the pendulum, and the butterflies ran to T cells, because if the antibodies don't work, we must go to the other arm of the immune system, because, dogmatically, we think it has to be an either or, but that's not how the immune system thinks. We created STEP, and STEP was clearly a disaster, right? We saw enhanced susceptibility, and those were vaccinated because we were activating the CD4 T cells. Again, the pendulum swung, right? Instead of really thinking through, it's not that we don't need T cells. T cells are important. We just need to have backup mechanisms that the immune system has evolved over millennia, right? Our antibodies and our T cells work together, right? And then we started learning about the innate immune system as if this was like a new discovery. Well, no, the innate immune system has been with us before we were even like diversified as mammals, right? And so all these components of the immune system work together, and learning how to make vaccines that can use these instead of having the butterfly of our community jumping from one to the other because one did not work properly, but understanding the system, understanding how the immune system really is able to maximally recognize, control, and clear pathogens, right? This is really, I think, at the essence of understanding how we can, get our community to sit still, talk to each other, work together. As somebody said in the chat, in the Q&A, we have to stop working as silos, right? This is where the success of COVID-19 really came into play, where everyone came to the table and began to share data in real time to try to advance the field as fast as possible, right? This is not a competitive exercise. This is an exercise of collaboration where we all come together, bring our best ideas, and make a big difference by thinking about the immune system as a collective. And so I think that breaking these dogmas of the either ors, or my favorite cell, or your favorite antibody, or your favorite technology, that has to be dropped at the table, and folks have to really begin to talk to each other. And I think we saw it happen. Now, the question is, how do we engage and continue that by trying to create these collaborative frameworks as opposed to silos, which is what we often do with funding pots, right? We create silos not, because we do it intentionally, but because we think that competition will breed innovation. And that's not the way I think we're going to get to the end of this pandemic.
Great. Other thoughts?
There was actually the question of the experimental medicine vaccine trials. Any thoughts on those?
I will. I think it's a really necessary step, because with all these modifications that will inevitably be necessary, the iteration has to be much faster. I mean, if we only look at a few things at a time, this could take many, many years, and I think that's unacceptable. The good thing is mRNA is one platform. There obviously will be other approaches that can also speed this up. So I think the experimental medicine framework is really important. I want to go back to also what Bill mentioned earlier. Bill mentioned ATI, or antiretroviral treatment interruption, in terms of a way to look at, treatment of people that are infected. I think one thing that I've seen brought up before is that the ATI framework can also be a way to evaluate, say, BNABs, broadly neutralizing antibodies. And because a lot of these trials that we've been trying to do in the past have taken a long time, but with an ATI framework, we could see if we have a good correlative protection or a correlative risk, in this case, a correlative protection. We can use the ATI framework to kind of screen through different potential BNABs. And then which BNABs are best, or which combinations are best, can also be applied to the vaccine development. So I did want to just add that, because Bill, when you mentioned ATI, that reminded me of that point as adding to the experimental medicine.
Yeah, I think it's too high of a bar. I think ATI is not really a good measure of what it would take to protect one virus from coming into a body after someone's already been infected. So I think we have to take that with a grain of salt, though. Anything we could do that would help with the cure effort would be useful. But I would like to remind everyone who remembers it, or tell the people who weren't there, that at the HVTN meeting a few weeks ago, Mark Harrington raised the question of what about manufacturing? And I think that besides having these small trials, which must be safe and people have to enroll in with the full knowledge of what they're doing, we need more experimental batches of immunogens. And Larry Corey, the ultimate person who's running this show, maybe not for good, is saying we need more things. If you talk to Bart, you hear the same thing. The money to make batches of vaccine and the wait lists for mRNA manufacturing is in our way right now. And it would not be an insurmountable investment to do some kind of a facility that would make experimental batches just the way the VRC does and the way the Chavis do for a lot of different experimental medicine trials. Because right now, that's what's holding people
But can I add, Bill? So, I mean, I think that it's not news, but I think all the big mRNA developers are moving towards, these self-contained, manufacturing sites that can be essentially planted anywhere. And so I think that is on the horizon. And I think that it's important, right, that manufacturing was a critical limitation, today, but it will not be in the future for all platforms. At least the mRNAs are incredibly flexible, very fast. That will not be the case for proteins or particles or other technologies where we do need to beef up and to, really make sure that we can get those products out there. But I think most important from the experimental medicine perspective, if I can just add one point, is that, we don't have mice, we don't have hamsters, we can't test concepts in small animal models. This is one of the biggest limitations we've had in the HIV vaccine development space. Monkeys are limited, right? We are not going to have enough monkeys to do enough testing. And of course, always this question about whether the monkey model is 100% faithful to the human model, right? Because of the differences in the virus and differences in the animal model. So learning how we can at least tune the immune system, right? Regardless of whether we're going to get efficacy signals out of there, but learning how to tune the immune system, we don't know how to do that, right? As we heard before, you know, we have some solutions that have worked for other pathogens, but the solutions for HIV will be different. And learning how to get durable immunity at the mucosal barriers where HIV enters, learning how to sustain those levels at the barrier, learning how to harness all arms of the immune system, not just the T cells or the antibodies, but really think of it holistically. Those are experimental studies that could happen today. And, you know, as we heard, there are clearly, you know, millions upon millions of individuals who would rise to the occasion to be part of defeating this pathogen. And so building up the capacity to even do those experimental trials, whether they're to end HIV or the next virus that enters into human populations, it's a gift to the world, and it's an obligation that we have as a scientific community.
I just want to add to that. First, Bill, let me counter what you said about ATI. I agree with you that at an absolute bar, it is a bar that's very high. But I think the point made earlier, and I think this was made by Shelley Karuna and Katie Barr, is that one can compare relative protection or relative diminution of HIV viral load. And I think in that way, ATI is useful. Jumping to the manufacturing, this is something, again, that Galit, as you said, I think there is an effort to expand manufacturing in different parts of the world. I think there has to be a lot more effort in this area. I think a lot of times, especially with US politics now, there's very much a focus on, you know, US based, but by improving the infrastructure, supporting groups like the Serum Institute of India and other, building up manufacturing capacity around the world will be a huge investment for everyone. And that is a big area. The last thing about experimental medicine, and this was also brought up at the HVTN meeting recently, is I think there is a real interest in a lot of low and middle income countries to do experimental medicine trials. One of the challenges right now with a lot of these trials is enrollment. A lot of the PIs in the US, in other parts, have mentioned that enrolling participants has been challenging. And I think if we can enroll in more sites, in more areas, there will be a lot more interest. And I know that Glenda Gray and Beatriz in Brazil really were pushing to get more involvement in these type of trials.
Let me just add, maybe you know, adding to final comments, since I see that we're almost at the top of the hour. So from the previous trials that we've done, large efficacy clinical trials that we've done, one of the things that has been evident is that there seems to be a shared vision amongst the communities and stakeholders whom we conduct these trials with in collaboration. And because of this shared vision and the interest in vaccine development, which obviously has come up through meaningful community engagement, at least in sub-Saharan Africa, we have that even if the results that have been received from current clinical trials, the ones that have just ended, even if they have been not as successful, we see that there's quite a lot of buy-in. And I would like to just say that, you know, because of this willingness to actually see vaccine success, to, you know, the desire to see vaccine success, it is important that even as we plan future clinical trials in the form of experimental medicine vaccine trials, it is important that this sort of engagement at all levels of community with regulatory bodies actually continues. That is going to definitely help us with recruitment. It's very important. The other thing I would also like to just mention is that in all the clinical trials that have been done, there's been a demonstration, and it's also been a learning point actually, that there's an impressive capacity that has been built at clinical trials in conduct of clinical trials in the very high standard and good retention that has been seen in all clinical trials and adherence. So I feel that these things that have actually, we've built momentum. It will be important that we're actually able to, continue on this trajectory, even as we plan upcoming clinical trials. So I just wanted to add on to some of the things that we have learned. And also the other important thing related to COVID-19, I wanted to just mention is, when you discover a successful product, it's not necessarily that it's going to be implemented. We've had a very low uptake of COVID-19 vaccine trials. And when I think about it around HIV vaccines, even if we were to develop a successful vaccine, will it be taken up? So the continuous community engagement, the continuous engagement of different stakeholders, I think will be key alongside our efforts to develop and find an effective vaccine. Thank you.
Thank you, William. And we are over time. We were going to kind of go around and hear final thoughts from everyone, but I think that those were really great final thoughts. And to me, it feels like the key message coming through to me is collaboration, which I think has always been the message in this field. But I think that we're at this time point, and all of you really put such nice texture to this, such nice detail about this, the place that we're in currently, and that we need actually even a new level of collaboration moving forward. And so I think as AVAC, as advocates, we're excited to work on that too. And I am going to actually hand over to our fearless leader, Mitchell, who opened the series on May 18th to just close us out.
Great. Stacy and Ethel and Galit and Dale and William and Bill, or Bill and William, or two Williams. Thank you for just such a remarkable conversation. And you saw, hopefully, if you haven't, you'll go back and look at the chat, for such love and praise for all the things that all six of you said. And it's impossible to summarize it all, both this call and the last 3 weeks, or last 3 calls in 2 weeks. And I will just say that I want to particularly thank the 6 of you, because when we embarked on this odyssey of 3 webinars in less than 2 weeks around HIV Vaccine Awareness Day, we got a lot of question marks, like, is there that much to say about HIV vaccines? We don't have one. It's hard. What more do you need to do and say than that? And you all gave proof positive to the fact that this conversation is far from over. And I'm so glad that we embarked on the 3 calls. We could have 3 more because you all showed the richness and diversity of perspective and all the things that we need to do. I think, as you probably saw, as someone chatted earlier in the call, this really is a roadmap, an advocacy agenda for the way forward. And you really put the exclamation point on this series. And again, it's impossible to summarize it all, but I will just say the richness of quotes from this call, certainly breaking the dogma. And Galit, I'm so grateful that you said it, because we do swing like this pendulum, or like the drunk butterfly, and we've got to get our act together as a community. And so I'm grateful for that. I also love that you said, sit still. You're clearly the parent of small children. But to have this field sit still and dissect what we do and don't know and plot the way forward. A number of us tried to say that in the commentary I put there at the end. And Dale, it really was remarkable to watch. I think every government employee should leave government service and get as unplugged as you just did. It was so, not that you didn't say brilliant things when you were in service at the NIH as an employee, but hearing you capture that as an advocate. And it's a reminder that whether you are a basic scientist, a clinical trialist, an advocate, we're all advocates, or we all need to be advocates. And you really, the comparisons of where we put not just money, but where we put our time and our effort really underscored that. William, I am so grateful because you kicked the session off so brilliantly about what's different about HIV, but you also reminded us at the end that this is not just about clinical trials, and that Africa is not just a place to conduct clinical trials, but you and colleagues have really shown what leadership in science looks like from all over the world. I hope Nita Marara, I'm not sure if she's still on, but asked a great question in the Q&A about examples of collaboration and global, regional, and local. And I think you really captured that. And I'm really grateful that you hit that last point about all of this work to just get us to interesting science isn't enough. We're here to get us to products. And I think all of you really captured that in the call. And Bill, at one point you did say you're now going to become a serious advocate. I don't know what you've been doing for the last 27 years, but you have been that serious advocate and reminded all of us at AVAC of why this work matters and really helped us conceive this session. So I just look back on this last 70 minutes and am just in awe of what we have accomplished as a community, but also inspired by the work we have to do to go forward. And as Stacy said at the very beginning, we had 4 Ps. We actually had 5 Ps. We didn't name the 5th P, but it was partnerships. And as Stacy rightly said, this has been about collaborations between AVAC and IAVI for many, now 2.5 decades, about working through our coalition to accelerate and support prevention research supported by USAID, the CASPER program. There's no one in HIV. The letter 1 doesn't appear. There's no one modality, vaccine or antibody or small molecule or microbicide. There's not one intervention. There's not one approach to an HIV vaccine. And there's certainly not one organization or any one leader, as you alluded to, Bill, that does it all. And we all know that. We don't need HIV Vaccine Awareness Day on May 18th to tell us that. But I'm glad we took the time out these last 2 weeks to remind ourselves of what matters. And I am deeply inspired by what all of you said today and the challenges ahead of us and the opportunities to work with a speed and an urgency that we did see in the last 2.5 years in COVID. And Galit, you've said it in different settings over the last couple of years, but I hope people are listening. It can be done. And as you said, I mean, I love your enthusiasm, the science that happened in the last 2 years. You couldn't have imagined this, and yet it happened. And it is that reminder that this is not a dream. These are things that we can make real when we work collaboratively. So all of that to say, I am enormously grateful. Ethel and Stacy for remarkable moderation. I want to thank Stacy and Bill for really pulling this whole series together. And you know, one other thing, you know, we always say that, you know, May 18th is just a day. It was a great speech by Bill Clinton 25 years ago at Morgan State University. And I'm a sucker for commencement speeches. It was a great one. But it's not about a single day either. And it's not even about a single series over 2 weeks. This is an everyday job that we all recommit to on May 18th each year, or today on May 31st. So I want to thank you all. And we want to keep hearing from you. Many of you have stayed on now for well over 2 hours, and thank you, or well over 1 hour. Thank you. Keep the questions coming. Don't wait till May 18th, 2023 to figure out what clever letter we're going to choose for next year. Be in touch day to day, because there is stuff that doesn't wait until 2023. So thank you all for your time, your efforts, and for your partnerships and your friendships. Because if you can't do this with people that you enjoy to be with, this work is even harder than it is on its own scientifically. So thank you all. Stay safe and well, and hope to see everybody in a place together sometime soon. Cheers all.
Bye all. Thanks again.
Bye.